One are men over 50, why this one ancient move restores testosterone overnight. The biology explained. Introduction. The allure of the overnight fix. The internet is awash with headlines that promise dramatic physiological transformations from a single, mysterious intervention. Phrases like, this one ancient move restores testosterone overnight, are engineered for clicks, sharing, and algorithmic visibility. They tap into a very real, very human desire, the hope that decades of hormonal decline, fatigue, diminished vitality, and age-related changes can be reversed quickly, safely, and without pharmaceutical intervention. For men over 50, the topic of testosterone is rarely abstract. It touches energy levels, muscle mass, sexual function, mood, cognitive clarity, bone density, cardiovascular resilience, and overall quality of life. When a headline suggests that a forgotten movement from antiquity can recalibrate this master hormone in a single night, it's understandable that curiosity turns into urgency. But biology does not operate on internet timelines. Hormonal systems are governed by intricate feedback loops, cellular machinery, genetic expression, and environmental inputs that unfold over hours, days, weeks, and years. Testosterone is not a switch that flips on and off. It is a dynamic steroid. Hormones synthesized through a multi-step enzymatic cascade, regulated by neural and endocrine signaling, distributed through binding proteins, and utilized by androgen receptors that must be transcriptionally active to exert physiological effects. No single posture, stretch, lift, or breath pattern can bypass these biological realities to restore testosterone overnight in men over 50. That said, the underlying curiosity in such headlines is not entirely misplaced. Certain movement patterns deeply rooted in historical physical practices do exert measurable influence on the neuroendocrine system. When performed with proper intent, loading, breathing, and recovery, they can improve circulation, reduce sympathetic overdrive, enhance insulin sensitivity, support healthy body composition, modulate cortisol dynamics, and create conditions that allow the hypothalamic pituitary gonadal (HPG) axis to function more efficiently. Over time, these adaptations can contribute to healthier baseline testosterone levels, improved androgen receptor sensitivity, and better symptom management for age-related hormonal shifts. The overnight claim is a myth. The physiological rationale for movement-based hormonal support is scientifically robust. This article will dismantle the sensationalism while honoring the legitimate science beneath it. We will explore how testosterone is actually produced, regulated, and utilized in the male body. We will examine what happens to the endocrine system after age 50, separating normal aging from pathological decline. We will investigate what ancient moves might actually refer to, tracing their historical context, and translating traditional movement wisdom into modern exercise physiology. We will review what peer-reviewed research actually says about exercise and testosterone, distinguishing acute hormonal fluctuations from chronic adaptations. We will explore the nervous system's role, the impact of stress and sleep, the influence of nutrition and body composition, and the environmental factors that modulate hormonal health. Finally, we will synthesize this knowledge into an evidence-based, realistic, and sustainable framework for men over 50 who want to support their hormonal health through movement, recovery, and lifestyle optimization. The goal is not to sell a miracle, but to provide clarity. You will leave with a deeper understanding of your biology, a realistic timeline for adaptation, and actionable strategies grounded in endocrinology, exercise science, and aging physiology. The truth is less dramatic than the headline, but far more empowering. Consistent, intelligent movement, paired with recovery and metabolic health, can meaningfully influence your hormonal environment at any age. Part 1. The biology of testosterone. How it's made, regulated, and used. To understand why no single movement can restore testosterone overnight, we must first understand how testosterone is actually produced, regulated, and utilized in the human body. Testosterone is not merely a muscle hormone or a sex hormone. It is a foundational steroid that influences virtually every tissue in the male body, from the brain and bones to the cardiovascular system and immune function. Its synthesis and regulation involve multiple organ systems, enzymatic pathways, feedback mechanisms, and cellular receptors. Let's break down the biology step by step. The hypothalamic pituitary gonadal (HPG) axis. The command center testosterone production begins not in the testes, but in the brain. The hypothalamus, a small region at the base of the brain, continuously monitors the internal and external environment. When it detects low circulating androgen levels, metabolic stress, or other regulatory cues, it releases gonadotropin-releasing hormone, GnRH, in a pulsatile manner. This pulsatility is critical. Continuous GnRH exposure actually desensitizes the pituitary gland, which is why pulsatile delivery is essential for healthy hormonal signaling. GnRH travels through a specialized network of blood vessels called the hypophyseal portal system to the anterior pituitary gland. There, it stimulates the release of two key gonadotropins, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. LH is the primary driver of testosterone production in men. It enters the systemic circulation and travels to the Leydig cells located in the interstitial spaces of the testes. Leydig cells and steroidogenesis. From cholesterol to testosterone, Leydig cells are the primary testosterone-producing cells in males. They contain abundant, smooth endoplasmic reticulum and mitochondria, organelles specifically adapted for steroid hormone synthesis. The process begins with cholesterol, which is either synthesized de novo within the Leydig cells or imported from circulating lipoproteins, primarily LDL. Cholesterol is transported into the inner mitochondrial membrane by a protein called STAR, steroidogenic acute regulatory protein. This is a rate-limiting step in steroidogenesis. Inside the mitochondria, the enzyme CYP11A1, cholesterol side chain cleavage enzyme, converts cholesterol into pregnenolone. Pregnenolone then moves to the smooth endoplasmic reticulum, where a series of enzymatic transformations occur. 3-VHESD converts pregnenolone to progesterone. CYP17A1 converts progesterone to 17A hydroxyprogesterone, and then to androstenedione, DRE. 17-VHSD converts androstenedione into testosterone. Testosterone is lipophilic, meaning it diffuses easily out of the Leydig cells and into the bloodstream. Once in circulation, approximately 98% of testosterone is bound to proteins, roughly 40–50% to sex hormone-binding globulin, SHBG, 50–60% to albumin, and only 1–3% remains unbound or free. Only free testosterone and a portion of albumin-bound testosterone that can readily dissociate is biologically active and able to cross cell membranes to bind to androgen receptors. The androgen receptor, where testosterone exerts its effects, testosterones. Physiological actions are mediated by the androgen receptor, AR, a nuclear receptor found in nearly every tissue. When testosterone enters a target cell, it binds to the AR. In many tissues, particularly the prostate and some brain regions, testosterone is converted by the enzyme 5A reductase into dihydrotestosterone, DHT, a more potent androgen that binds the AR with higher affinity. In muscle and bone, testosterone itself often acts directly. The testosterone AR complex translocates to the nucleus, binds to specific DNA sequences called androgen response elements, AREs, and modulates gene transcription. This process influences protein synthesis, cellular differentiation, mitochondrial biogenesis, insulin sensitivity, erythropoiesis, and neuroplasticity. Importantly, androgen receptor sensitivity varies by individual, age, genetic background, epigenetic modifications, and lifestyle factors. Two men with identical testosterone levels can experience vastly different physiological and symptomatic outcomes based on receptor density, post-receptor signaling efficiency, and tissue-specific enzyme activity. Circadian rhythm and pulsatile. Released testosterone is not secreted at a constant rate. It follows a distinct circadian pattern, peaking in the early morning hours, typically between 6 a.m. and 8 a.m., and reaching its lowest point in the late afternoon or evening. This rhythm is regulated by the suprachiasmatic nucleus, SCN, in the hypothalamus, which synchronizes hormonal output with light-dark cycles, sleep architecture, and metabolic states. Additionally, LH release is pulsatile, occurring roughly every 1 to 3 hours in healthy young men. These pulses ensure sustained Leydig cell stimulation without receptor desensitization. As men age, both the amplitude and frequency of these pulses can diminish, contributing to the gradual decline in testosterone production. Why overnight restoration is biologically impossible. Given this intricate biological framework, the claim that a single move can restore testosterone overnight defies basic endocrinology. Consider the following realities. Steroidogenesis takes time. The conversion of cholesterol to testosterone involves multiple enzymatic steps, mitochondrial, transport, and cellular signaling cascades. These processes cannot be instantaneously upregulated to clinically meaningful levels by a single physical maneuver. HPG axis. Feedback loops are conservative. The body tightly regulates testosterone through negative feedback. Elevated levels signal the hypothalamus and pituitary to reduce GnRH and LH secretion. Acute spikes are quickly normalized.